Oh, okay. Well, I guess we are
Yeah,
live.
yeah. Can you hear me?
Yeah, I can hear you. Always perfect. So, welcome everybody. I hope you are enjoying your evening on this hot day that the sun of the Netherlands had to offer us. Uh, so, I'm Margarida. Before I forget to introduce myself, um, yeah, and the other host of this evening is going to be Guilherme. Hi, Guilherme. How are you? I cannot. It just okay. Guilherme is
Hi
there.
everyone. Yeah, hi Mida. I'm good.
Yeah, nice. Shall we toast to
Yes.
some kind of yeah, just cheers before
Cheers.
make first to our invited speaker of today. So actually, today we have invited speaker. Her name is Caitrin Crudden. She is from Ireland. She did her master studies in Manchester in the UK. Then she traveled a bit further and she went to Sweden to do her PhD in Karolinska Institute. Afterwards, she decided to try and see the Dutch culture. And so she came to Amsterdam, where is she now doing her research as a postdoc in UMC Amsterdam. And yeah, she's going to introduce us now. Hey, Katrine!
Hello.
Hello. Welcome.
Thank you
Yeah.
very much for having me. This is nice. Yeah.
Well, thanks for accepting the invitation.
Yeah, absolutely. It's nice to have something new to do in lockdown. I'm pretty bored of my four walls.
Yay! Yeah.
Cheers
Well, cheers, Katrine.
for another round of Pint of Science.
Yes. So yeah, Margarita already very kindly introduced me. My name is Caitrin Crudden, and I work as a postdoctoral researcher here in the Netherlands at the Amsterdam University Medical Center. And I work as a cancer cell biologist. So today I want to tell you guys about my favorite subject, cells. Why, and a little bit about why on earth we would want or care about understanding them, and give you a little bit of a hint about the research that I do. And then, of course, please feel free to ask me whatever questions that you'd like. So I guess that I'll start with the basics. So you guys and me are made up of many parts that need to work in an incredibly coordinated and highly synchronized fashion in order to keep us healthy, alive, drinking beer. And we can think of the cell then as the smallest unit of this biological life. So we have somewhere, although it's quite difficult to quantify exactly, we have somewhere in the range of around 30 trillion cells. So trillion. So you can begin to imagine the kind of complexity that a system must need to try to coordinate the action of trillions of independent units. So, I mean, how on earth would this happen? I mean, it must do, but how? Well, cells talk, they chat, just like you guys very kindly brought me on here for a chat. They talk endlessly, and they talk in their way. They give out information from one cell in the format of molecular messages that can enter the bloodstream and travel around to different parts of the body. Another cell then can take in this message, can read it, and can respond in an appropriate way for the betterment of us. But I mean, it sounds quite simple if I say it like this, but I know you guys work in biology and in medicine and think that you guys understand that it's phenomenally complicated. And it gets a little bit more difficult because what we're thinking about, if we go scale-wise, an average cell is somewhere around the range of around 10 micrometers. So if you can kind of remember your ruler at school, and you have your centimeter, and then you have your little millimeters, in that little millimeter gap, you can fit 100 cells side by side. So you can really understand the scale that, you know, this communication needs to take place on. And if, for example, I have, you know, a cell in my finger that wants to communicate with a cell in its pal, a cell in my big toe, that's a pretty extreme distance for, you know, a message to go across. And well, I know Margarita, but I'm not tall in the Dutch scale of things, right? Actually, I'm quite tall in Ireland, but not so much here. So for a tall person, the distance between for a finger cell and a toe cell becomes astronomical. It's also not just the distance that they need to communicate over. If you think about it, if you're the size and the scale of a cell, and you give out one of these molecular messages, out there in the body, in the bloodstream, is basically like the Wild West for a cell. There's fluids rushing around, you're getting battered and bombarded and squeezed and ripped apart. And basically, tiny, delicate molecular messages ultimately, very often, would just get destroyed in this system. So I came up with a bit of an idea to describe it. So if we try and think of a cell communicating in that way, sending out a molecular message to its friend, you can think about it a little bit like if you want to write a letter to your cousin in Australia, right? So this metaphor is pre-Facebook times. So you get out your nice stationery, right, that you got for Christmas, and you'd write a lovely little letter. And if you were a cell sending out a molecular message into the bloodstream, this is a bit like me taking my beautifully scripted letter, opening the window and throwing it out into the wind, and hoping one day my luck might come in and the wind will take that letter and drop it onto my cousin's doorstep in Australia. I mean, it might happen, but it's not a particularly efficient way of communication.
So like throwing a message in a bottle in the sea and hoping it gets to the proper person.
Right. I mean, it's certainly, why not give it a go, right? People do it. It works. But it depends how important, I would guess, that your message is, right? Is that, do you want to send one message out in the hope, or what actually happens in those kind of cases? So this does happen, for example, hormones and things like that. But what you need to then do is completely flood the system. So you need to send out millions of replicate messages in replicate bottles if you really want to get your message across, right? So it's not really efficient that way. But, yeah.
Okay. Shall I let you come to the butt, or should I ask you something?
Yeah, you go.
Yeah, nice. I was actually wondering if indeed in most cases there's this bottle in the sea drawing away. And I was wondering if there is like some kind of way that you can make a message come through the recipient. If there's a way that cells, because I mean, I can imagine that sometimes cells communicate a bit, try to ask, I don't know, maybe for help, or just they are bored and maybe they want to chat, no clue, like us maybe like us, but maybe if they want to be maybe more specific, is there a way to do it?
Yeah, that's a really good point. Yeah, so if we think about what I talked about in writing a letter and throwing it out the window, right? So that can be a metaphor for if a cell just throws out a message into the bloodstream, hopes maybe it gets to its cousin. But you're right, there is a more efficient way to do this, right? If we think back on our scale, what do? We don't throw letters commonly into the wind, but we do something else that works. So for example, right, you guys very, very kindly sent me this lovely little glass, right? Look at that. You sent me this in the post, right? And today I get to have it safely and securely and enjoy it in a little beer guzzling way, right? And I precisely get to enjoy this lovely glass because you guys wrapped it very nicely. You put it in a protective box. And then, just as you said, Margarita, you wrote my name and address on it, right? So my neighbors didn't steal it. And it got to me. It's here. And it's really appreciated because it's a very hot day out there in Amsterdam. So a cold beer is much appreciated today. But this is what we do, right? We don't throw letters into the wind. And funny enough, this is exactly what cells do. So cells also send packages, protected packages, out. And this is a much more efficient way of communicating. So cells give out little boxes, little packages that we call exosomes. Now, not only do the exosomes contain that message, so whatever you wanted to write in your lovely little letter, they often actually contain multiple messages all put in together. They're wrapped up nicely, so they're protected. They're in an outer package, in this case made of lipids, fats. And just as you said, the clever thing is, very often they have information on the outside about the recipient. So it actually gets to its intended place. Pretty clever.
So one quick question, perhaps. How often and how does the body deal with like a mislabel? Sometimes I may write the wrong zip code, for example, in a letter. Is this common to happen in our bodies, and how does the body deal with that?
Yeah, I guess there's certainly sometimes when it's just general uptake. So there's certainly some cells that are not differentiating, are not only taking in things that are addressed to them, that we have some cells that are very ET cells, as we call them, like phagocytic, and they're really engulfing everything. That's their job. So you have to send out a lot of packages with the right address to be sure that it really gets there. So it's only really in certain circumstances where you can really put an address on and it gets to that particular cell. In reality, it's a lot more messy. It will go to lots of different places, maybe that have similar addresses, or they could be generally taken up by different cells. So it's a little bit of a mix. It's not as perfect as maybe PostNL is here and delivers quite a lot of our packages quite well. It's a little bit of a mix, but it certainly is a lot more efficient of a communication and a long distance communication system than, you know, just throwing out those naked molecules into the bloodstream and hoping for the best.
Yeah, and actually in a similar vein, we have a question from Hannah and she's asking if cells struggle with something similar to language barriers. How different type of cells, how do they communicate, and if that is complicated for them?
Do they struggle with language barrier? Well, I would say in general, actually, biology has quite a surprising commonality. So it kind of, you would think maybe that, okay, yeah, your liver cells speak in a different language, you know, than your cardiac heart cells. But the weird thing is, a lot of the kind of machinery that we're talking about here, the machinery that's packaging, that's creating this cell postal service, is not only happening in every organ in our body, it's happening in every animal, it's happening in very simple organisms. So to be honest, it's actually usually these kind of systems are quite well conserved. By that we mean they probably they were developed and generated quite early in the evolutionary timeline, and therefore they're actually surprisingly similar throughout the organism, and at least there's a similar toolbox that a lot of them use. So that's what I would say. I think that there's kind of surprisingly a common language, I would say.
That's very interesting.
Yeah, quite funny. Actually, we have another question from JM. So, what kind of message do cells send to each other? And in other words, what would the finger cell want to tell to a toe cell?
I mean, I can only guess what my finger wants to say to myself. Yeah, that's a good point. So, in lots of different situations, I guess the messages are going to differ. So, if we talk about normal physiology, it's always for the overall goal, this communication of keeping you healthy and keeping everything balanced and working perfectly. So, for example, if I had a paper cut on my finger, I'm not going to demonstrate. If I had a paper cut on my finger, then the cells in my finger need to scream for help, right? They are destroyed. They're chopped in half. They need help. So they scream out for help. And those messages that flow from that area are what's attracting all of these helper cells. So be that a helper cell is away doing a different job in my toe, it suddenly hears the scream. It needs these helper cells need to come to the area that needs help and do all of the things that are needed in that area. They need to help, stop clot the blood, stop blood loss. They need to stop germs from getting in. They need to start going about all this a highly coordinated process whereby we can now grow cells and repair that injury. So it's very situation dependent, but those are the kind of things. They cry out for help when something needs repair. Sometimes things need to grow. Sometimes cells need to die for the betterment of an organ. So it's about communication so that everybody knows, okay, we need to do this thing so that, she's all right, those kind of things. It's all for the, hopefully, at least in normal physiology we're talking about here, we can go into disease. But in normal physiology, that's usually what the communication is. What do we need for the greater good, i.e. you, me, whatever body they're in.
Nice. And I guess so then what happens in a disease situation then? What goes wrong? Does they give the wrong name to the package, or what does happen then?
Yeah, so this is a great question, and I'm glad you brought it up so that people are not asking themselves at home, why on earth are we spending our Wednesday evening listening to somebody talk about the cell postal system? This does not seem very important. I'm here to tell you it is. So it's important for exactly this reason, is that I work in cancer research personally, and what goes wrong in cancer is that this normal cellular communication goes wrong, right? So cancer cells are not this kind of, alien pathogenic entity that comes in and does something in your body. Cancer cells start off as your normal, healthy body cells, but something goes wrong and they turn rogue. And what happens is that when cancer cells have started, they no longer participate in this harmonious communication network that we're talking about, that always is for the good of the whole body. So one of the first things that cancer cells do is they stop listening. So this back and forth communication where we need to know what is appropriate, they stop listening to the messages that tell them when it's appropriate to grow, and more importantly, when it's not appropriate to grow. Cancer cells are not listening to that. First of all, they stop listening. Secondly, they then eventually change so that they're actively sending out rogue information. And that's why we, in particular, are interested in this cell postal system, because what cancer cells end up doing is that they send out these packages, these exosomes, and they contain cancer-promoting information. And when those packages now are taken up by normal healthy cells, that information manipulates them. And this manipulation helps the disease to progress, allowing, for example, metastatic spread throughout the body. And unfortunately, I think we're all then painfully aware of the mortality that is associated with this kind of situation, with the progression of the disease throughout the body.
So in a sense, let me see if I understood correctly.
Yeah.
They basically stop listening and start spreading misinformation.
Bingo.
I know a few politicians like that also. But interesting. So would it be possible to, in a way, attach a signal or a marking in this rogue information? So to tell the body, like, stop listening to this. This is labeled as like rogue information. It's not important for us.
Well, certainly, if that could work, it would be very, very helpful. One of the problems in one of the issues of why treating cancer is so difficult and why cancer research is a very complex field is that to directly target something, you want your target to be sufficiently different than all of the things that you don't want to hit, right? And cancer cell is very difficult to then hit. It's not a pathogen. It's not something that looks completely different, a virus, a bacteria. It looks remarkably similar to our other cells. And therefore, the problem is both the cells themselves, but also those packages that they're given out, look remarkably like the packages that should be in the body's postal system. So the immune system finds it really, really difficult to separate those out. So certainly if we can get, and if we can generate and we can reactivate the immune system to recognize these as rogue communication packages, shouldn't be there. Absolutely, this could be one way of getting the immune cell reactivated to recognize the cancer cell. And this, there's a lot of research going on. It's called immunotherapy, and it's all about reactivating the immune system to recognize the cancer cell and killing those cancer cells like it does so incredibly efficiently with, you know, any sort of pathogens, usually when they get into our body. So yeah, certainly if we could do that, that would be an incredibly strong therapeutic strategy. It's very tricky, kind of hiding in plain sight.
All right, thanks.
Nice. Caitrin, can you tell us a bit also more about what you are doing in your research? What are you focused on? Do you focus on one of these ways of stop cancer through this message, or how you do it?
Yeah, great. So yes, what I'll do very first before I go into what I do, because then you can see it with your own eyes, is I'm going to show you a very quick video that helps to explain this. I'm going to show you this because, firstly, why otherwise would you believe me and my rambling? But we as science lovers should always look for the evidence, right? So this cell, what you're looking at here is a single cervical cancer cell. And every time you see a tiny flash of light, that cancer cell is giving out one of these packages. So some of my colleagues developed this really cool tool to allow us to see with our own eyes and the help of a microscope. We're able to see the exact point where this cancer cell is giving out those packages, right? I think it's very obvious, right, to the eye. I hope you guys can also see that. And
it's pretty great.
Yeah, it's nice. It's quite beautiful, isn't it? If you don't think of it as a horrible, what it could be doing. Actually, you don't have, in particular, you don't have to worry about this cell. This cell, you know, lives in our lab. Don't worry about it. It's fine. But what I hope that you guys can get from this video is the idea of, you know, how many of these packages, these rogue packages that every cancer cell is giving out. So if you imagine, you know, a tumor with thousands, maybe millions of these cells, you can imagine if these are inside a patient, just how much rogue information is flooding the system. So that's what we're dealing with to begin with. So to answer your question, Margarita, there's loads of things actually that you can do with these exosomes, and that's why we focus on that. So I work in a group called the Exosome Research Laboratory. We're situated here in Amsterdam at a center called the Cancer Center Amsterdam. And our job, me and my colleagues, is we study this exosome-mediated communication, the cell postal system, if you will, and we try to figure out ways that we can use it, manipulate it, hijack it for patient benefit. So there's lots of things you can do. Me personally, my research is looking at, I'm trying to discover new drugs that can really stop this method of communication. So I want to find out new ways where I can limit the cancer cells' access to the postal system. If I can do that, the cancer cell at an earlier stage can no longer, adapt and hijack, manipulate the rest of the body. We can hopefully stop the disease progressing into those more dangerous stages and hopefully save lives. So that's one thing. That's what I focus on. Let's just stop this. Let's stop this crosstalk. But there's actually loads of really cool things that you can do with the cancer cell postal system. So some of my other colleagues, for example, so if we know that cells are using this method of communication, and in a patient, they're everywhere, okay, can we read it? We know the cells can read the information, but can we, as cancer researchers, as clinicians? So can we take a simple blood test from a patient and look for these cancer exosomes? If we can detect them, we can learn a lot more about what's going on in the body. And you can imagine we could diagnose cancers a lot earlier. That absolutely save lives. We could just learn more about the disease that's going on in this patient. And the more we can understand about everybody's individual case, the better we are at using the therapies and strategies that we have in a better way. The other really cool thing that we can also do is, okay, we know that these packages are being sent around. Can we manipulate them? Can we put something in the packages? They already have addresses on them. They already have nice little biological protective packages. We know it works. What about if we put a drug in there? We write the cancer cell's address on it. We send in the chemotherapy, for example. It goes directly into the post box of the cancer cells. It doesn't go to neighbor cells, the healthy cells. They don't get the toxic drug. It doesn't have their address on it. In that way, if we could do that, if we could deliver drugs in this way, we could use our current therapies better, make them stronger, and have absolutely fewer side effects, which for some of the very old therapies that we have, these are huge issues. So there's a lot of cool things you can do with the cell postal system.
Yay!
That's really great.
Yeah, sounds very cool and very great. I mean, how did you end up doing research in this topic, in this specific?
I think that I've always been interested in cells, I guess, and biology in general at school. And I went and I'd done quite a broad undergraduate. And I was drawn to cancer research genuinely because of the complexity. I mean, it's kind of one of those things that we've been studying intensively for decades anyway, very intensively in the field. And it seems from the outside, before you go into it, we're like, are we making any progress? I mean, what's happening? We are making a lot of progress, but it's very, very complex. And yeah, it's a minefield in there. So I think I got drawn to cancer cell biology for the complexity. I also think cells in general are cool. So cancer's a really interesting way to see, okay, this is how normal cells work. These are the intricate differences that go wrong. And astronomically wrong. They can have huge detrimental effects. We all know that. So I think that's how I got into kind of cell biology and cell communication. And then I worked what I was talking about earlier on with the throwing the letter in the wind. I actually done my PhD on something a bit more like that. So I done my PhD in what's called growth factor signaling. And growth factors often are these little naked molecular messages that are given out. And sometimes they don't spread, they don't need to spread to the other side of the body. Often it's kind of local, it's within the little community, the neighborhood of cells, and therefore they do a little bit better short distance. So I studied a lot about that, about the kind of cell-to-cell communication in the local environment and how these molecules, these growth factors, mediate kind of a cell growth and cell dysfunction in cancer. And then I wanted to change a little bit for my postdoctoral study. And yeah, this field of extracellular vesicles or exosomes is kind of been coming up in the last decade or so and starting to get really interesting. So it's what we like to call a hot topic. So it's quite attractive. Everybody wants to get into the hot, cool new thing, right? So I think I was attracted into that to stay within cell-to-cell communication, which I'm very interested in. And I think it's very valuable that we know about and just go a little bit different. And I think that's how I got into the cell postal system and its intricacies.
Oh, cool. Yeah, it sounds very interesting, for sure. I think the time flies and we are already half an hour talking, which is quite nice. And yeah, being great, it shows a lot of potential, how cells can talk and how can we use them.
Well, it was a very quick run through, but hopefully it gave you a little bit of an idea in a nutshell how cells might talk, and also importantly what we're trying to do to intercept that and to help cancer patients in the future.
Yeah, but just one last question: is this just happen to be more known to be in cancer specific, or it's also other diseases? Also, people are studying this kind of mechanism. I don't know, to find new drugs, new therapies.
Yeah, definitely. I'm talking very much from a cancer researcher perspective. That's my field of interest and that's my expertise and my background. But absolutely, this postal service works all over the body. It works in normal physiology without disease. And therefore, it goes wrong, or it can be intercepted or manipulated in a whole host of diseases. So it's absolutely not a cancer-specific thing. That's just from the perspective I come from. But there are lots of exciting avenues to develop it for either research into just how normal physiology works, which is 100% valuable without translation, but certainly in lots of other disease and interesting biological phenomena. Yeah.
Yeah, cool. So anything else you wanted to talk about, Caitrin? The floor is yours.
No, I don't think. I think we touched on pretty much everything that I wanted to talk about. Thank you very much for having me, guys. And I hope at some stage I can meet you and hopefully a lot of our viewers in where we're supposed to do this, down in the pub at some point. Maybe sometime in the future. I'll bring my own glass.
Yeah, bring us all. Yeah, we hope so. But it was great to have you here.
Thank you.
Yeah, it was a lovely talk. Yeah, I think that's how I guess you are going to finish, unfortunately.
Yeah.
Hopefully see you. Yeah, hopefully see you maybe another time than in person.
Yeah.
In a pub, maybe in Amsterdam, maybe somewhere else. But yeah, let's toast once last time. I don't know. I just feel like we have the cups and we can just.
Yeah. Thanks everyone for showing up.
Yeah.
Thanks for all the questions, and stay tuned for more events to come.
Yeah. Next one in 2 weeks. See you.
Bye.
Bye.